Today we're going to talk about bryozoans. Bryozoans belong to the group Protostomia, although there is still much debate between that and the deuterostomes. The consensus at the moment is they are part of the protostomes. They are also part of the group of Lophophorata. Inside that group, there are two other phylum, the Phoronidia and the Brachiopoda. And what unites the three phyla are the Lophophore, which is a ciliated feeding structure, and an epistome, which is a structure that helps move food from the Lophophore to the mouth. Along with that, all three phyla have an adult body that possesses three distinct parts, each of which with their own coelom. Classifying them, Bryozoa can be split into two main classes, the Gymnolemmata and the Phylactolemmata. The Gymnolemmata they house all the marine organisms as well as certain freshwater species, and can be further split into two subclasses, the Eurystomata and the Stenolemmata. In the Eurystomata, we have two orders that house the vast majority of all the bryozoans, the Ctenostomata and the Chelostomata. In our other class, the Phylactolemmata are all of our freshwater specimens. And as a whole, for bryozoans, there are up to 6,000 recognized species. Start with some general characteristics. All bryozoans are sessile zooid colonies. Of course, there are some few exceptions, being this biology, but as for growth forms, they grow as encrusting, branching, and gelatinous forms, and they can occur at all depths, anywhere from the surface to the ocean floor, and at all latitudes from the equator to the poles. They can be found in both marine and freshwater, and most reproduce via budding, and their sexual reproduction occurs via an ancestrula. As for the zooids, they consist of a polypide, a tentacle crown, and a gut. And then they have a cystid, which is their body wall. Moving into the zooids, they are what compose the colony of bryozoans. So a cystid is the outer body wall of the bryozoan and consists of living and non-living structure. It is what houses the polypide. And the polypide includes the Lophophore, which is the ciliated, and the tentacular feeding structure seen up there. Opening up the cystid, which allows the polypide in and out, is the orifice. Each zooid on a colony of bryozoans can have its own function. Most zooids seen are autozooids, which are really there for feeding, and that's about it. The heterozooids, there can be multiple different types, but the main three are kinozooids, avicularia, and the vibraculum. The kinozooids are very reduced, and they are modified for attachment to a substrate, like a disc, a holdfast, or a stolon. The avicularia are only found in the Gymnolemmata, and it is an operculum modified as a mandible, thought to be used to defend the colony or clear away the debris. And the vibraculum, as seen in this picture, are modified avicularium with a flagellar-like operculum. In a way, they are used to help some discoid chelostomes move around in their free-living lifestyles. As for their body wall, it is a large trait in using a taxonomic identification. Each different bryozoan colony will have certain specific species-specific spines, pits, and protuberances on their body wall. These can be used to determine species from other species, as well as function in defense of the overall body. To bring the Lophophore in and out of the cystid, they use a combination of circular and longitudinal muscles. It involves increasing hydraulic pressure in the cystid to move it up and down. When they decrease the pressure, the Lophophore is drawn back in. Along with their body wall, they have been found to house a lot of different symbionts or commensals that live within them. Dendy in 1963 found 5 main groups of protozoans, annelids, arthropods, flatworms, and nematodes, rather living inside or around them. As for feeding, the main feeding structure in the bryozoans is the Lophophore, and it differs depending on which class. So our freshwater group, the Phylactolemmata, have a horseshoe-shaped Lophophore, just like the one seen here. And their mouth is going to be found at the central location, moving down to the body. Our Gymnolemmata, the Lophophore is circular-shaped, again with the mouth in the center. However, the circular shape helps them feed because it forms a funnel shape around the mouth and allows the ciliated tentacles to beat and create a water current to bring food into the mouth. As a whole group, they are suspension feeders. However, the exceptions exist where their Lophophores have been known to reach out and grab larger organisms such as zooplankton and hold on to them. One study that examined their feeding pattern was Pratt in 2004 to determine whether or not the spacing between the zooids of a colony to determine how well it feeds and how efficient it is. And what he found was that when the zooids were spaced closer together, they were more efficient in feeding, which could be used to help increase their growth rates as seen in Membraniophora, and how it spreads so rapidly because it's a fast grower. Their digestion is very unique. So they have a U-shaped digestive tract, just like our crinoids we've seen, and it occurs extracellularly in the cardia, which is a small valve that goes through and leads into the central stomach. It occurs intracellularly in all parts of the stomach. So to start for the path of the food is drawn into the mouth via a ciliated current, and it rests on top. When the mouth opens, the food is sucked into the pharynx, where it then moves down into the cardia, where the extracellular digestion takes place. From the cardia, it will move into the descending stomach, and then into the central stomach, where all the intracellular digestion takes place and breakdown of it. After the central stomach, it moves up into the pharynx, and through the pharynx into the rectum, where in the rectum it can be transformed into things called brown bodies, which we'll get into in a minute. And then from the rectum, it is released out through the anus. As for the circulatory, gas exchange, and excretory systems, they are very simple. There is no real circulatory system. Any and everything occurs through diffusion. Any transport is done through coelomic fluid. Gas exchange is the same. There's no respiratory pigments in bryozoans, so they use diffusion. And just like it occurs across the lophophore due to the large surface area. As I mentioned earlier, the brown bodies, functioning with the digestive system, the brown bodies occur because there's no excretory system in them. So they compress all of the non-digestive particles and either render them inert or release them as a body. And the nervous system is again extremely reduced. All they have is a central ganglion up here next to the mouth, and then they have a large oral ring that goes around their mouth to spread out to the rest of the bodies. Sensory is mostly have a tactile sensors on their tentacles used in their feeding and used to expel waste. When they reproduce via sexual reproduction, all the colonies are hermaphroditic. However, certain zooids can be gonochoric, or certain zooids can be hermaphroditic, depending on the species. The initial stage of growth in all forms of bryozoans is the ancestrula, and it moves on to that. In the Phycolammates, they brood via invaginations of the body wall, which we'll get into in development. The tenostomes attach eggs to the atrial wall, and the chelostomes use an ovocell. An ovocell acts as a placenta in species with small eggs. This research was done to show the 3 different types of reproduction in the chelostomes. The main type we have, the first one is non-brooders, and these are where the zooids are produced in large batches of small eggs. And embryogenesis takes place without parental care in the seed. The non-placental brooders, the zooids produce eggs in small batches, and embryogenesis takes place inside a brooding space of the ovocell. In placental brooders, the zooids produce a small egg at maturity, and a zygote receives nutrients from the mother just like a placenta. The asexual reproduction is used mostly for colony growth and healing damage. It occurs via budding, and is unique in the freshwater bryozoans because they are able to produce statoblasts, which are a stress-resistant reproductive form that are released when conditions are not good. They can remain stagnant for long periods of time until conditions become well enough for them to grow again. Their development is unique to each group as well. So in the Phycolammates, they are again placental brooders getting the nutrients from the mother. The larva is ciliated with a short swimming phase before it settles and spends most of its time growing and settling, and they have an incomplete gut. In the Eurystomates, they use a laterally compressed siphonodes, as seen in the top right. Siphonodes are found mostly in the tenostomes and chelostomes, and they have a complete gut and can remain planktonic for months until they find an opportune place to settle. This is, before we finish up, a research that was done to do a survey of all the different species seen here, especially on the east coast of Florida. It was done in the Indian River area and found upwards of 66 species of chelostomes, 15 ctenostomes, and 3 cyclostomata, all in different types of habitats such as seagrass beds, intertidal and subtidal zones, as well as rock hash and sand. And to finish up, the ultimate question, can you eat them? Not really, no. There's nothing good to eat within them. A lot of them are just small enough that there's no nutritional value with them. So, thank you.